Welcome to Bio 211 Plant Physiology Unit 1, Session 6, Factors Affecting Photosynthesis and its Importance of Photosynthesis. By the end of this session, students should be able to list the factors that affect or influence photosynthesis, explain how each factor affects the process, explain what is a limiting factor, and show how important photosynthesis is to life. The term rate always involves time, so the rate of photosynthesis can be considered to be how fast photosynthesis can take place. This can be measured by the amount of glucose produced by a plant over a given time. This topic is especially important to scientists and farmers. By understanding the factors that affect the rate of photosynthesis, they can do work to try and increase the rate of photosynthesis to increase the yield of a crop. Before we account for the product, we need to consider first what goes into making that product. So we can state by looking at the summary equation: carbon dioxide, light, and water. Like all chemical reactions, a temperature component is required. So we add that and discuss each in turn. Three factors can limit the speed of photosynthesis: light, intensity, carbon dioxide concentration, and temperature. So light. Without enough light, a plant cannot photosynthesize very quickly, even if there is plenty of water and carbon dioxide. Increasing the light intensity will boost the speed of photosynthesis. The rate of photosynthesis increases when light gets brighter. If the rate of photosynthesis is plotted against light intensity, the graph shown in the figure below is obtained. So we have photosynthesis, the rate of photosynthesis against light intensity, and you see that it begins from a point and it does move to another, it moves steadily, and you notice that the rate of photosynthesis increases linearly with increasing light intensity, as you can see from the graph. So many plants spread out their leaves in such a way that each leaf maximizes the amount of light falling on them, and the lower leaves are not shaded by the ones above. That's leaf mosaic. So too much light at a high intensity can damage chloroplasts. Some woodland plants photosynthesize more efficiently in dim light and so are called shade plants. In the laboratory or even a suitable place or table in your house, you could set up an experiment to find out this for yourself. So where you have difficulty of space, get together in groups or assemble in the lab for this experiment. Get the following materials: a glass bowl or an empty transparent plastic gallon, a beaker empty, and that's about 300 milliliters Belaqua or 500 milliliters Voltic or any other bottle. Then a meter rule, a table lamp or bedside lamp. You have water, then we have some granite chippings or a sharp kitchen knife, submerge aquatic plant in a water bowl or petri dish, example Eloidea. So set up the experiment as follows: fill the glass bowl almost full with the water to create a water bath. You will need to cut off the top half of the gallon to get your bowl with a kitchen knife. Cut off the funnel top of the Belaqua bottle to get your beaker. Then fill 3/4 full with water. Drop the aquatic plant into the beaker with water. If you are using Eloidea, you need to hold the cut end up to the beaker. Put the setup of plants in beaker of water into the water bath. Water bath will create a constant temperature around the plant setup. So place a table lamp or bedside lamp on a table some distance from the water bath with the plant. So with the meter rule, you can vary the distance between the photosynthesizing plant and the light. Your setup should look like something like this. So in here you have a lamp which is being held by a clamp. Then you have a boiling tube, you have your Eloidea plant, then you have your meter rule which you're using to do your measurements, taking or recording your measurements. So if you had to use the water bottle beaker, you may have to hold it down in the water bath, placing some granite chippings or sand in the bottom before filling with water. Create a table for recording the rate of photosynthesis against distance of light from photosynthesizing plant. So you have a table like this, and that is how you're going to use it. How are you going to determine the rate of photosynthesis, whether photosynthesis is going on in the first place? The light intensity may be assumed to be equal to the changing distance between the photosynthesizing plant, or you may decide to calculate it using the formula below, where light intensity equals power over area, where the power is the wattage of the bulb giving the light, and the area is the measured distance from the photosynthesizing plant. Since the bulb throws light in all directions, the light of a sphere is assumed and used as such. The measured distance becomes the radius. The formula then becomes light intensity equals W over 4 pi r squared. So plot the light intensity against the rate of photosynthesis. Did your graph look like the one in figure 6.1 in your textbook? We hope it did. So the environmental factors affecting rate of photosynthesis. We are done with light. We are now looking at carbon dioxide. It is used to make sugar in the photosynthesis reaction. The concentration of carbon dioxide in the Earth's atmosphere varies from 0.03% to 0.04%. An increase in the concentration of carbon dioxide gives an increase in the rate of photosynthesis. It is difficult to do this out in the open air, but it is possible in a greenhouse. So sometimes photosynthesis is limited by the concentration of carbon dioxide in the air. Even if there is plenty of light, a plant cannot photosynthesize if there is insufficient carbon dioxide. When the rate of photosynthesis is plotted against varying concentrations of carbon dioxide, the rate of photosynthesis increases linearly as carbon dioxide concentration increases till further increases in the concentration of carbon dioxide has no effect on the rate of photosynthesis. So study figure 1. A to B shows the linear increase in the rate of photosynthesis, and B to C shows the plateau point when the rate of photosynthesis becomes constant regardless of the further increase in carbon dioxide concentration. What would account for this? So as we discussed in the figure, many crops such as tomatoes and lettuce give higher yield when grown in greenhouses. Farmers add additional carbon dioxide into the greenhouse to increase the concentration, and so the rate of photosynthesis of the crops. The additional cost of the carbon dioxide is worthwhile because of the yield, the increased yield. Some companies have used this to great environmental use. Rather than pump waste carbon dioxide into the atmosphere as a pollutant, they redirect it into big greenhouses where plants such as tomatoes use it during photosynthesis. This not only reduces their carbon footprint but gives an additional profitable product. Temperature. The higher the temperature, typically, the greater the rate of photosynthesis. Photosynthesis is a chemical reaction, and the rate of most chemical reactions increases with temperature. However, for photosynthesis at temperatures above 40 degrees Celsius, the rate slows down. This is because the enzymes involved in the chemical reactions of photosynthesis are temperature sensitive and destroyed at higher temperatures. To better understand the effect of temperature on photosynthesis, it is important to know the effect of temperature on the enzymes involved in photosynthesis. Enzymes are affected a great deal by temperature. If the temperature is too cold, the enzymes move around too slowly to meet the substrate and for a reaction to occur. As the temperature increases, though, so does the rate of reaction. This is because heat energy causes more collisions between the enzyme and the substrate. However, as you will remember, all enzymes are proteins, and at too high temperatures, the proteins break down. The active site of the enzyme becomes distorted, and so the substrate no longer fits, and hence the reaction does not occur. We see that the enzyme has been denatured. Greenhouses are used to capitalize on the effects of higher temperatures, increasing the rate of photosynthesis. Plants from regions of warmer climates can successfully grow in colder regions by using greenhouses. The rate of photosynthesis does not increase with higher temperatures for all plants. Plants which grow in colder climates have an optimum rate of photosynthesis at low temperatures. Therefore, different types of plants have optimum temperatures of photosynthesis. So you have a rate of photosynthesis and the optimum temperature. You realize that you have a bell-shaped graph. So as you can see, when the rate of photosynthesis is plotted against temperature, there is a steady rise in the rate of photosynthesis till a point is reached when the rate of photosynthesis is optimal. Beyond the optimum temperature, which is about 25 degrees Celsius, in this particular instance, there is a steady decline in the rate of photosynthesis to zero at 40 degrees Celsius. So what could account for the decline in photosynthesis at higher temperatures beyond the optimum temperature? Go over the discussion on temperature if you are unable to answer straight away. Limiting factors of photosynthesis. In 1905, when investigating the factors affecting the rate of photosynthesis, Blackman formulated the law of limiting factors. This states that the rate of physiological process will be limited by the factor which is in shortest supply. Any change in the level of a limiting factor will affect the rate of reaction. So in a process like photosynthesis, which is affected by more than one factor, its rate is limited by the factor which is closest to its minimum value. So at any point in time, if one of the three factors is in low supply, this factor will be the limiting factor. Only a change to the limiting factor will increase or decrease the rate of photosynthesis. Changing the other two will have no effect. So for example, the amount of light will affect the rate of photosynthesis. If there is no light, there will be no photosynthesis. As light intensity increases, the rate of photosynthesis will increase as long as other factors are in adequate supply. So as the rate increases, eventually another factor will come into short supply. The graph below shows the effect of low carbon dioxide concentration. So you have higher carbon dioxide concentration, that's higher temperature. Higher carbon dioxide concentration, you have lower temperature. Lower carbon dioxide concentration, you have lower temperature. That's rate of photosynthesis against intensity of light. So limiting factors of photosynthesis, it will eventually be insufficient to support a higher rate of photosynthesis, and increasing light intensity will have no effect. So the rate plateaus. If a higher concentration of carbon dioxide is applied, light is again a limiting factor, and a higher rate can be reached before the rate again plateaus. If carbon dioxide and light levels are high, but temperature is low, increasing temperature will have the greatest effect on reaching a higher rate of photosynthesis. Farmers can use their knowledge of these limiting factors to increase crop growth in greenhouses. They may use artificial light so that photosynthesis can continue beyond daylight hours or in a higher than normal light intensity. The use of paraffin lamps inside a greenhouse increases the rate of photosynthesis because the burning paraffin produces carbon dioxide and heat too. In addition to the three main factors above, other factors such as chlorophyll concentration, specific inhibitors, water and pollution that affect photosynthesis. To these we now turn our attention. As we noted above, photosynthesis may be affected by other factors. Their effects may not be limiting, but certainly do have negative effect on photosynthesis and so bring about low yields in plant produce. These factors are chlorophyll concentration, water, pollution, specific inhibitors, and mineral deficiency. Without plants, life as we know it would not exist on our planet. Green plants play a vital role in the following areas: atmospheric gases. During photosynthesis, plants take in carbon dioxide and give off oxygen as a byproduct. Photosynthesis can therefore be considered as the real phase of respiration. Without green plants performing photosynthesis, there would be no way for nature to replace all the oxygen being consumed in processes such as respiration and combustion. Furthermore, the levels of carbon dioxide in the atmosphere would increase. Thus, the balance of atmospheric gases is kept stable by photosynthesis. Food. Green plants are called producers because they produce all their own food from the raw materials around them via photosynthesis. Animals and humans, on the other hand, are consumers, and all the food they eat comes directly or indirectly from plants. Most of the world's population obtains more than 80% of their food directly from plants. For example, rice, potatoes, wheat, corn, etc. The remaining source comes from animals, and these animals are part of the food chain, which always begins with plants. Clothing. Many plants have cells that are long and thin with thick cell walls. These properties allow the cells to form fibers, which can be spun and woven into fabrics. Examples of these include the cotton plant, used to make cotton, and the flax plant, used to make linen. Medicines. Plants contain a vast range of chemicals which are extracted and used in the production of medicines. Aspirin, the drug used as a pain reliever and to reduce blood clotting in heart patients, is derived from salicylic acid, a chemical extracted from the bark of the willow tree. Much stronger painkillers and analgesic drugs such as morphine and codeine are produced from opium, the dried sap derived from the seeds of the poppy plant. To date, the number of plants tested for medicinal properties number only in their thousands. There are still a vast number yet to be tested, including many of the species sourced from the tropical rainforest. The unknown medicinal properties of these plant species adds to the importance of protecting natural habitats such as rainforest. So, in review, we have considered both man-made, environmental, and structural factors that affect photosynthesis. We have also noted how carbon dioxide, light, and temperature may play limiting effects on the rate of photosynthesis, and concluded the session by showing how important photosynthesis is to both humans and all other objects.